welcome to the class of quality assurance, and this subject is in the syllabus of B Pharm course in 6th semester, and the code of this subject is BP 606T, and today we are going to discuss about another very important topic that is given in the unit 5 of quality assurance, that is general principles of the analytical method validation. So, this is another very important topic, and that is very relevant to the whatever the things that are happening in the pharmaceutical industry, especially in the perspective of the quality control and method development. So, without going to without wasting our time, let us start. I am Dr. Alankash Srivastava, Associate Professor in Kite Group of Institution, Ghaziabad. So, first of all, what is the objective of analytical method validation? The objective of the validation of an analytical procedure is to demonstrate that it is suitable for its intended purpose. So, you can easily understand there are various different types of analytical methods that are given in the pharmacopoeia also. There are many different sources of getting the reference of these analytical methods. For example, there are many journals which are publishing exclusively different types of analytical procedures for various different kinds of analytes. So, first of all, what is the objective of this method validation? The first objective is to ensure whatever be the reading that you are getting through applying your analytical method, whether it is fit for its purpose or not. So, analytical methods, whether it may be any kind of a titration simply, or whether it may be a very complicated procedure, for example, liquid chromatography coupled with mass spectroscopy, these all analytical methods needs to be validated or revalidated before their introduction into the routine use. So, for another question arises, what is this analytical procedure? So, analytical procedure or analytical methodology that refers to the way of performing the analysis of any analyte. Suppose you want to perform the analysis, suppose you want to perform the analysis of paracetamol tablet in the tablet dosage form. So, what you have to do? You have to select the suitable analytical method through which you can perform your analysis. So, this is analytical methodologies basically are the different types of ways for performing the analysis of any suitable analyte. And another thing, it should describe in detail the steps necessary to perform each analytical test. Now, analytical methods needs to be validated, as well as there may be a chance that it requires the revalidation. What are those circumstances in which analytical methods needs to be validated or revalidated? The first thing, before their introduction into routine use. Now, second thing, whenever a condition change for which the method has been validated. For example, an instrument with different characteristics or samples with different matrix. Suppose you have performed the analysis in tablet, now there is a change in the formulation, now you want to perform the analysis in the capsule dosage form. Both dosage forms are different. So, in this case also you will have to validate your analytical procedure again. So, there is another case, or there may be you have another instrument, but now you have procured another instrument with having a more advanced or better characteristics or very different characteristics of performing analysis. That means, again you will have to revalidate or validate your methodology. So, whenever the method is changed and the change is outside the scope of the method, there you will have to validate the analytical method. So, if you have changed the method in some way, that means, and again another that means you will have to go for revalidation again. Now, the question arises, what are the different types of analytical procedures that are requires validation? Now, the discussion of the validation of analytical procedures is directed to the 4 most common type of analytical procedures. What are these different types of procedures? The first kind of analytical method is the simply identification test. Identification tests are also a type of analytical procedures which are commonly used. Another type of analytical methods are quantitative test for impurities content. So, in this case we are actually not dealing with the drug content, we are not actually determining the drug content, we are simply determining the level of impurities that are present in our dosage form or matrix. So, this is another type of analytical method which requires validation. And the third thing is the limit test for the contents of the impurity. There are various different types of limit test also, for example, limit test of chloride. So, these all limit tests are also requires validation. The next type of or the last type of analytical procedure which needs validation or it is essential to perform the validation studies, that are quantitative test of an active moieties in samples of the drug substance or drug product or other selected components in the drug product. So, these are the 4 types of analytical procedures or methodologies which requires validation before its actual usage. Now, what are the different validation characteristics? So, these all are the different validation characteristics for validation of any analytical method. The first parameter is accuracy, then precision. The precision is further divided into 2 types, that is repeatability and intermediate precision. Then specificity, detection limit, quantitation limit, linearity and range. So, these all are the validation characteristics that are to be performed for any validation for any analytical methods before using it for its actual use. Now, furthermore, revalidation may be necessary in some circumstances. Suppose you have already validated an analytical procedure, but now there may be a chance that after some times you require revalidation of any analytical method. So, what are those kind of circumstances? The first circumstances may be there may be a changes in the synthesis of the drug substance. So, if there is a change in the pathway of the synthesis of any drug substance, that means there may be a chance that there may be a different types of impurities that may come along with the drug. So, that means there is a change in the matrix, there may be a there may be a probability of getting some different types of impurities along with the drug. So, there is a possibility or there is a this is one of the circumstances in which you will have to revalidate your analytical method again. Now, second thing, there may be a change in the composition of the finished product. So, if you have changed the composition of the finished product, for example, you are previously preparing tablet and later on you have gone to prepare the injection, that means the both formulations having a very different matrix. That means you will have to revalidate your analytical procedure again. And the third thing is the changes in the analytical procedure. So, if you have changed the analytical procedure, for example, previously you have adopted to perform the analysis of your analyte using titration, but later on you started using UV spectrophotometry. That means there is a change in the analytical procedure, and obviously in this case you will have to revalidate your procedure again. Now, the degree of revalidation required depends on the nature of changes. So, what is the degree of revalidation that actually determines on the basis of what is the nature of changes that you have done in your these all things. So, these are the cases in which we have to perform the revalidation. So, first of all, the first parameter for the validation of analytical method is specificity. Now, what is this specificity? Specificity is the ability to assess unequivocally the analyte in the presence of components which may be expected to be present. Now, typically these might include impurities, degradants and matrix. So, in this test or in this parameter, actually what we are doing, we have to confirm that the whatever be the response that we are getting through our instrument by our analyte, that should be from our analyte only. There should not be any merging of responses along with impurities as well as any degradant or the matrix. That means the whatever the response that we are getting is false. That is not because of the truly that is not because of the truly that is not truly because of the analyte. So, these are the cases, and because of this, the first parameter that we are actually performing for a validation of analytical method is specificity. The method should be specific. The next thing is the accuracy. Now, accuracy of an analytical procedure expresses the closeness of the agreement between the values which is accepted either as a conventional true value or an accepted reference value and the value form, and this is sometimes also referred as trueness. As you can see in this figure, you can see all the arrows heated in the same area, that is the very close to the target. That means these arrows are actually these arrows are heated the accurate target. So, in the same way, all kind of analytical method should be accurate enough, or it should show the true value of the analyte, and that is actually accuracy, and that is why it also sometimes terms as the trueness of analytical method. So, I hope you have understood about the what is accuracy. Now, the next thing is the precision. Now, the precision of an analytical procedure expresses the closeness of the agreement, that is the degree of scatteredness between the series of measurement obtained from the multiple sampling of the same homogeneous sample under the prescribed conditions. The precision may be considered at three levels, that is repeatability, intermediate precision and reproducibility. So, first of all, the question arises, what is the difference between accuracy and precision? Accuracy, that means the closeness of the values to the true value, and here you can see through this figure there are three different figures. Here you can see all the arrows miss the target, this is the target, and all the arrows miss the target, and here you can see all the targets are all the arrows heated this shield in a scattered way, and that is why these can be not precise because all the readings are scattered. So, these are not precise, neither accurate, because they all have missed the target. That means, these are not precise, that are neither accurate. But in this case, the arrows almost hit at the same place. That means, there is a very less or negligible scatteredness. That means, the overall things are precise, but it is not accurate because no arrow actually hit at the target. But and also in the third figure, as you can see, it is precise and accurate both, because all the arrows heated into the target and the results are not scattered, and that is why this is precise and accurate both. So, in this way you can easily understand the same thing about the analytical methodology. So, if the analytical procedure is overall accurate, but it is not hitting the actual target value in a very scattered way, it is actually hitting the target value in a very scattered way, that means the method is not precise, and this precision should be established before proceeding for the uses of the analytical procedure in actual use. So, this is the difference between, as you can easily understand through these figures, what is the difference between precision as well as accuracy. Now, there as you can as you have already seen in the previous slide, this precision is having the 3 levels. The first is the repeatability. So, what we are doing in this repeatability, basically in this case we are actually we are actually performing the method over a short interval of time in a repeated manner, and this repeatability is also termed as intra-assay precision. So, within a very short interval we are performing the repeated experiments, and we are actually here calculating the deviations that we have seen through this experiment, and if the deviations are within the specified limit, that means our method is repeatable. And the next level of the precision is intermediate precision. So, in this case we are actually performing the analytical method in different laboratories, in different days, or different analysts and different equipment. So, if the methodology is correct or suitable enough, that means there may be no deviation between the results, whatever be the time that we are performing the experiment, or whatever be the different equipment, or there may be a different analyst. So, if the deviations are within the limit, that means the precision or intermediate precision is enough or suitable for the said analytical method. Then the next level is the reproducibility. So, in this case reproducibility basically expresses the precision between the laboratories, that is collaborative studies usually applied to standardization of the methodology. So, what we are doing in this case, we are changing the laboratories. So, suppose if you have changed the laboratory, that means that the overall all the things the analyzed as well as the days and the timings or the environment, all these things are changed, but even in this case also, if the analytical method is producing the same result, that means your method is reproducible at any laboratory. So, these all are the different levels of the precision. The next parameter is the detection limit. So, what is this detection limit? The detection limit of an individual analytical procedure is the lowest amount of analyte in the sample which can be detected, but not necessarily quantitated as an exact value. You can easily understand all the analytical procedures are having some particular limit of having or of detecting an analyte, but here you can see that detection limit is that concentration of the analyte which can be detected, but it is not necessarily quantitated as an exact value. So, this is actually the detection limits definition. So, by detection limit by establishing the detection limit, we are actually establishing the amount of analyte that can be detected by your analytical methodology. So, if the analytical procedure is that simply means the analytical method is not suitable for use if the amount of analyte is below the detection limit in the matrix whatever you are going to detect. So, these are actually the detection limit, and the next type of analytical procedure or next type of the validation is the validation parameter is the quantitation limit. Now, what is quantitation limit? The quantitation limit of an individual analyte procedure is the lowest amount of analyte, is actually the lowest amount of any analyte that is present in the sample which can be quantified, which can be quantitatively determined with a suitable precision and accuracy. So, if any analyte is there, that can be the minimum amount of analyte that can be quantified by a suitable precision and accuracy level, that means that the analyte is reached is quantitation limit. So, now through these lines you can easily understand the value of detection limit will be always lower than the quantitation limit, because in this case we are the analytical method is having is reached to its maximum capability of just detecting the analyte that is present in any matrix. But in this case we are actually capable this is actually the estimation of the capability of the of any analytical methodology to quantify the amount of analyte that is present in the sample. So, these are the two different parameters which are also used to establish for in the validation of analytical procedures. The next part is the linearity. Now, what is the linearity? The linearity of an analytical procedure is its stability is its ability within a given range to obtain test results which are directly proportional to the concentration or the amount of analyte in the sample. So, now you can easily understand with the increase in the concentration of the analyte, there should be a increase in the response of the response from the detector in a same way. So, linearity basically establishes the response that is receiving in the detector or of any instrument is directly proportional to the concentration of the analyte. And for the establishment of linearity, a minimum of 5 concentration is recommended and other approaches should be justified. Now, the next part is the range. The range of an analytical procedure is the interval between the upper and lower concentration amount of analyte in the sample, including the concentration for which it has been demonstrated that the analytical procedure has a suitable level of precision, accuracy and linearity. Now, you can easily understand that every reading that is showing by the instrument may not having a sufficient level of the precision, accuracy and linearity, and the range is actually that range in which the analyte can be determined with a sufficient level of the precision, accuracy as well as linearity. For example, the assay of the drug substance or the finished drug product, it is normally from 80 to 120% of the test concentration. So, this is the range. Then next thing is the robustness. The robustness of the analytical procedure is the measurement of its capacity to remain unaffected by a small but deliberate variation in the method parameters and provides an indication of its reliability during normal usage. For example, if we are determining the analyte using HPLC or high performance liquid chromatography, we can change the mobile phase in a very deliberate manner or a very small proportion, and we will measure the change in the response. So, if the response is within the specified limit, that means the method can be used in spite of some very few changes. And now, the last part of these slides is the validation characteristics. So, now you can go through this table and you can easily understand what are the different parameters that needs to be established before proceeding before establishing the before usage of an analytical method. So, in this case there are three things. The first thing is the identification. Identification method may be there. There may be another type of method, that may be a impurity determination method. The impurities may be quantitative, or there may be a limit test, or there may be a assay type method. So, these all are the 4 different types of method that we have already discussed in the initial slides. So, these all are the parameters which are required to be established for these respective methods. The minus sign shows that these parameters are not required to be established, whereas the plus sign shows that these parameters should be established. For example, specificity should be established for all the kind of these analytical methods, whereas accuracy is not required to be established for the identification methods. So, this is all about the validation of the analytical methods. So, hope you all have liked this video. Thank you, friends.